All right, next part of the story, we're going to talk about this cross bridge cycle, which we've already talked a little bit about before, but we're going to go back over it again. So we have finally gotten to the point where our actins and our myosins are coming together, they're bonding to each other, and they're going to slide across each other, and that's going to allow muscle fiber contraction to take place. So that's our cross bridge cycle. Now, as long as you have calcium present, you have high intracellular calcium levels, you're going to be able to do the cross bridging. Okay, you've got to have that intracellular calcium level high in order for actin and myosin to come together and make their connection. If you don't have a high level of intracellular calcium, then the tropomyosin, that ribbon, is going to slide back over those binding sites, and actin and myosin are not going to be able to make their connection. So we've got to keep our calcium levels up if we're going to keep contracting a muscle. And what causes that? Well, you got to have action potentials spreading across the sarcolemma and triggering that calcium to come out of the SR, or the sarcoplasmic reticulum. So cross bridging again is when we have an energized myosin head. Remember, it has to be cocked like a trigger and contacts actin, and then it fires or does its power stroke to allow the sliding to take place. Pulls the thin filament toward the center of the sarcomere. Again, the sarcomere is the subunit of a myofibril. A myofibril is a long chain of a bunch of these sarcomeres, these little contracting units that you have. Let's see, come on, slide advance. There we go. All right, we've got some words here writing out what's going on. I'm going to go ahead and flip over to the diagrams here because it's easier for you guys can read the writing, use that as part of your notes. But I'll just review what happens during the cross bridge cycle here. All right, so what we're doing, we're zooming in on here is a 2D representation of a sarcomere. And again, in blue, this will be the center of the sarcomere. And over here, you've got those Z lines. Those are the borders of the sarcomere. And then in blue, you've got your thin filaments. In red, you've got your thick filaments. And so we're zooming in on an area where one thin filament and one part of a thick filament are coming together. All right, so our tropomyosin here has been moved out of the way. You can see you've got calcium ions attached to tropomyosin. They're in yellow. So tropomyosin has been moved out of the way, and our myosin heads, because of that, are able to contact the actin. And also notice at this point you've got an ADP and an inorganic phosphate. ADP plus PI stands for inorganic phosphate. That's a phosphorus bonded to 4 oxygen atoms, and it's an inorganic molecule, so sometimes it's abbreviated as PI. Where do these come from? Those are generated when adenosine triphosphate, or ATP, is split into those two molecules, like we talked about when we were going over cellular respiration. Okay, so if you guys remember, when that happens, when ATP gets split, that releases energy. That releases energy. And in the case of our cross bridging here, that's where the energy comes from that cocks the myosin heads. It pulls them back into an energized position. So, and we talked about that in an earlier video lecture. But so starting the story here, we're starting from that point. We've got the cocked myosin heads, and they have attached to the actin filaments. Okay, and then we move on to the next step, the power stroke. So you guys can see the myosin heads go like that. And as they do that, that drags the thin filament toward the center of the sarcomere. Okay, so over here, on this side to the left, the thin filaments are being dragged that way, and over here on the right, the thin filaments are being dragged that way toward the center. So you have this happening, again, all over the place inside that muscle fiber. You've got tons of these in those three-dimensional sarcomeres all over the place. notice your ADP and your P come off. So ADP and inorganic phosphate are released, and that change is called the power stroke. Okay, next step, if we're going to keep sliding, we've got to have our cross bridge detach. All right, and so in order for detachment to occur, ATP has to bind the myosin heads. You've got to have ATP molecules doing that, and that makes the myosin head detach. All right, and then continuing our story over here. Now we have ATP hydrolysis. Okay, and ATP hydrolysis is just where we go from ATP. We split that molecule into two smaller ones, ADP and inorganic phosphate. That releases energy. Hydrolysis means water splitting. You add a water molecule to ATP and that splits it in half into ADP plus phosphate. That's what hydrolysis means. That releases energy. When we break bonds, that releases energy, and that energy causes the myosin heads to go into the cocked position. So we have cocking of the myosin head, kind of like you're cocking a trigger. All right, once that happens, now we go back up here. We're back up at the beginning. The myosin heads are going to attach to the next actin binding site. Okay, and then they're going to go through their power stroke again. And so the actin filament is going to be dragged a little bit further this way. ATP attaches, myosin heads come off. Myosin heads come off. Oops, looks like my pen has shut off here for a second. Myosin heads come off. ATP has attached, it hydrolyzes, it breaks apart into ADP and phosphate. Boom, you recocked the myosin heads, and they bind to the next location. And so this just keeps happening over and over and over again. That's why it's called the cross bridge cycle, because it'll keep happening over and over and over again as long as we need to contract that muscle fiber within our muscle. Okay, so that's actually, finally, we've reached the end of the muscle contraction story. There's a lot of physiology in there, and I know it's kind of a heavy-duty topic, but if you just kind of work your way through the story and practice with it, it's not quite as monstrous as it may seem. But just remember, this story started back up when we generated our action potential on the motor neuron, and that led all the way to our cross-bridging cycle that we're mentioning here in this video lecture. That cross-bridge cycle will continue as long as you have high levels of intracellular calcium. As long as you're maintaining that, you're going to keep doing your cross-bridging. What happens to the calcium when you're no longer going to keep the muscle fiber contracting? The calcium has to be bottled back up into the sarcoplasmic reticulum. It actually gets pumped back inside there. Okay, so we're going to keep doing this cross bridge cycle that you see here over and over and over again as long as calcium levels are elevated. All right, and what keeps those calcium levels elevated? Action potential. As long as you have action potentials spreading across the sarcolemma, you're going to keep those calcium ions elevated. All right, so what this means is in order for a muscle fiber to keep contracting over and over and over again, like if you want to generate a fairly strong contraction, that means the motor neuron that's controlling it has to keep sending action potentials to signal it, hey, keep contracting, keep contracting, keep contracting, keep contracting. That keeps your calcium ion levels elevated. Now when you're ready to stop contracting this muscle fiber, the motor neuron quits sending its signals. So what's going to happen? You're going to actually wind up with lower calcium ion levels, and the reason for that is they get pumped back into the SR. All right, so as soon as you lose that signaling, those action potentials, the calcium ions start getting pumped back into the sarcoplasmic reticulum. They get bottled back up pretty quickly. Once your calcium ions get bottled back up in the sarcoplasmic reticulum, guess what happens? Your tropomyosin ribbon slides back over the binding sites on the actin filaments, and you break your cross bridges. So you've got to keep those calcium ion levels high if you want to keep running your cross bridge cycle and continue with your contractions. The bottling of the calcium ions back up, I referred to that as pumping. So that also uses a lot of ATP. So earlier during the course, we heard about how important ATP is as an energy molecule, a fuel molecule, and we've seen several examples here. You need ATP for running those sodium-potassium pumps in the nervous system to reset membranes. Those are going to be important in your muscles as well. You've got to have ATP to make your myosin heads come off from the actins, and then those have to get broken. Those ATP molecules get hydrolyzed to provide the energy to cock the myosin heads. And now we're seeing that ATP is used as a source of energy to pump calcium back into the SR where it needs to be stored, so it's poised and ready the next time you need to start contracting this muscle fiber. So ATP is extremely, extremely important for these muscle fiber contraction processes. All right, we have finally reached the end of kind of the nitty-gritty, detailed part of muscle physiology. In the next lecture, we're going to wind up starting to discuss more about what's going on in the muscle as a whole, and that'll bring us into muscle mechanics. And so that'll be the subject of lecture number 12 for chapter 9.